their samples. So we are going for it that, okay, which like one kind of organisms, or maybe fungal infection, or maybe anything, any microbe is there, or fungal isolates will be there. So we are doing it. It does at present this research is going on. So we are just trying our best. So we want to find out that, okay, is there any in future, is there any like infection maybe spread, or any kind of outbreak, or if any drug resistance organism we can find out. So we are like doing that. So I'm just doing it with one of my students. Maybe he will just go outside India after that this work. He's trying also. So we are working here. Even we don't have that much facilities here, but I am trying to collaborate with PGI microbiology lab. So we can just identify the organism there with the help of MALDI-TOF or with the other techniques. So just we here only just culture, and we can see which which species is there. Species level identification can be done later on. We just see it like it's organism, fungal, microbes, or whatever we will get.
Hello.
Okay.
So first of all, I request Usha Shri, you introduce again to the meeting, and then everyone will introduce in short, in 2 minutes only. Everyone will introduce, including Dr. S.W. Kulkarni will also introduce himself. First, Usha.
Sir. Yes, sir. Good morning, everyone. I'm Dr. R. Usha Sri, working as an associate professor in Department of Microbiology, Justice Bashir Ahmed Syed College for Women, Tenampet, Chennai. I'm very glad to invite you all for this graceful occasion scientific event. I would request one by one to introduce themselves to this scientific event. Okay. Thank you so much.
Dr. S.W. Kulkarni.
Yes. May I introduce myself?
Yes.
Yes, sir. Yes, sir.
Yes. Very nice to meet you all online. I am Dr. S.W. Kulkarni. I am a retired professor in microbiology, having 38 years experience as a teaching and nearly about 25 years research experience. I have completed my PhD under guidance of Dr. M. Deshmukh sir. So I'm very proud of him. And I am associated with the Microbiology Society starting from its establishment, that is 1996. And we are doing many things such as workshops, seminars, conferences, national level, international conferences we have conducted. Also, we conduct many courses for the students. MSc students. Yes. Regarding actinomycetes, I have completed my PhD on actinomycetes itself under the guidance of Dr. Deshmukh. May I give brief introduction of actinomycetes, please, sir?
No. Only that later on. First, only introduction.
Okay. So I completed my PhD under guidance of Dr. Deshmukh on soil actinomycetes from Solapur districts of Maharashtra. And I have so many papers on my name with Dr. Deshmukh also. We have written some books also. And I'm very glad today to have introduction with all of you and have associated with all of you. Thank you.
Yes. Then one by one you take the name. Usha Shri.
Yes, sir. Dr. Aparna Kumari. Dr. Aparna Kumari.
Good afternoon, ma'am. Actually, I'm not Dr. Aparna Kumari.
Okay.
I'm Aparna Kumari now. I'm pursuing my PhD.
Yeah. Kindly introduce, ma'am. Kindly introduce yourself, ma'am, to this gathering.
Yeah, sure, ma'am. I'm Aparna Kumari. I'm pursuing my PhD in VIT Veloor campus. and I'm interested to know this actinomycetes topic. So I joined this meeting, and I know this is very helpful for me because many of expertise people are there in this meeting. So I just want to know about this. Thank you.
Okay, thank you. Dr. Sunil Kumar, sir, kindly introduce yourself. Sir. Dr. Sunil Kumar, sir, are you here? Sunil
Yes.
Kumar, sir?
Yes, ma'am. I'm here. I'm sorry. So presently I'm working in Chandigarh University in the Department of Medical Lab Technology. So we are working here to isolate the organism or actinomycetes species specific from like people who are working in the laboratories. We are taking out their
Okay.
samples, swab samples. So
Okay.
one of student is working on that. So let's see if any species or anything will come out from those cultures or not. So we are working on that. It's totally like a new area here. Maybe the people are not worked in this area. So in nearby area, the labs are there, people are working. So we want to isolate these things from the people who are working in the labs. So this is presently we are working on.
Thank you so much, ma'am. Dr. S. Saravanan, sir. Dr. S. Saravanan, sir. Could I request you to speak? Saravanan, sir. Are you available?
Go to next.
Dr. Sunita.
Yeah, good afternoon, Deshmukh sir, Kulkarni sir, Ushashri madam, and all the other delegates present here. I'm Dr. Sunita Pundale, and I'm working in Hislop School of Biotechnology, Hislop College, Nagpur. I am interested in actinomycetes. I have been working on actinobacteria. I prefer to call them actinobacteria. I have been working on actinobacteria since for the past 15-20 years. I have completed my PhD on actinobacteria, particularly Streptomyces genus. And after that, I've shifted to rare actinobacteria. I have already completed one DST-sponsored project on rare actinobacteria. And currently, I am again, I have been awarded a DST-sponsored project on rare actinobacteria. So glad to be here and talking to all of you who are interested in actinobacteria, sir.
Thank you, ma'am. Komal Chowdhury. Miss Komal Chowdhury, could I request you to introduce yourself? Dr. S. Saravanan, sir. Okay. Meenakshi Agarwal. Miss Meenakshi Agarwal, could I request you to address the gathering? Miss Meenakshi Agarwal, she left. Komal Chowdhury.
Hello, good afternoon, everyone.
Yes. Yes, ma'am.
Hello, I'm Komal Chaudhary. I'm from Ahmedabad. Currently, I'm doing my PhD on bioprospecting of marine actinomycetes, and I'm working as an assistant professor in MG Science Institute, Ahmedabad, ma'am.
Okay, ma'am. Thank you so much. Saravanan, sir, are you here?
Okay, let it be, let it be. There may be some
Okay.
audio problem. So let us, again, I'm requesting Dr. S.W. Kulkarni.
Sir, still people are joining, sir. Shall I request them?
Okay.
Sivakumar, sir. Mr. Shivakumar Kannan. Mr. Shivakumar Kannan. Miss Loveleen. Loveleen, could you please introduce yourself to this gathering? Loveleen. Miss
What you can
Swati.
do? There are audio problems, it seems. So let us start with Kulkarni, sir. Dr. Kulkarni,
Yes, sir.
sir. in 5 minutes, you focus on actinobacteria. That's a very true. We'll call it as actinobacteria.
Correct, correct.
Yes. Dr. S.W. Kulkarni within 5 minutes.
Okay, okay. Thank you, sir. Thank you very much. Thank you for giving this opportunity on giving my opinion on actinomycetes as such. So actinomycetes is a group of bacteria, no doubt. Other than these two bacteria, we know, but they are having a they are playing very great role in environment, and therefore they are very important. Basically, the actinomycetes, they are known for the production of antibiotics. Antibiotics so far known to the human beings, out of that, nearly about 90 to 95% antibiotics, they are produced by the variety of actinomycetes or actinobacteria. Mainly streptomyces, they are involved in the production of antibiotics. They produce antibiotics which work against the bacteria, which work against the fungi, which work against the viruses also. So antiviral, antifungal, antibacterial antibiotics, they are produced by these actinomycetes as such. Apart from this antibiotic production, many other co-metabolites, they are also produced by the actinobacteria. In actinomycetes, we are having many genera. The major is the streptomyces, because streptomyces, they cover nearly about 70% of the actinobacteria. And in streptomyces, number of species so far discovered, 100 together species, they are known of the streptomyces genera. Apart from this streptomyces, there are other genera also. For example, Nocardia, Streptoverticillium, Micromonospora, Micropolyspora, like that. So many genera also there, and under every genus, there are many species so far discovered. Even though some species they are discovered to date, but still there is a wide scope to discover. So we can say only 20% species they have been discovered so far. So much more scope is there for the discovery of all these species from this particular variety of genera from the actinobacteria. Actinobacteria, they occur naturally in many sources, such as they are predominant in soil, they are predominant in water. In water, you can isolate from fresh water, you can isolate from the sewage also, you can isolate from the marine also. So like that, variety of the species you may get. They are known to cause some diseases also. They are known to cause some diseases in plants, in human being also. But apart from this particular ability to cause a disease, the major stress we have to give on the utility of the actinomycetes, actinobacteria as such, because they are known for that. They are eco-friendly, and many activities are there which they perform in environment. Actinobacteria, they produce number of enzymes which can degrade much more complex substances, complex compounds, which are not easily degraded by other microorganisms. So therefore, this is a very great achievement and great role the actinobacteria they play in degradation of many complex substances, and they make them available to plants, other community, microbial community in the nature, and therefore they are playing a very key role in the environment. They are known for antibiotics, already I have told you, enzymes, other metabolites, for example, melanin. So melanin is a particular product, pigment produced by this Actinobacteria, which is very helpful. Microdopamine is also produced by this particular Actinobacteria. So many products are there we are using nowadays. They are helpful. Actinobacteria, they are having role in industries, variety of industries. They are having role in medical branch, in agricultural branch. Therefore, these Actinobacteria, they are playing a key role, we can say, in the environment. And therefore, they are useful. And therefore, more attention have been given on the research on the Actinobacteria. The main thing in carrying out the research on Actinobacteria is the particular worker working on Actinobacteria requires a lot of patience. This is because they are slow growers, we can say, in comparison with other microorganisms. But once you have the particular growth, and when you are habituated to grow these particular Actinobacteria in circulation, in continuation, there is no much more time will be waste. Only initial, you require some patience. Once you got the trick, once you got the knack about the particular media which to be used, the cultivation conditions to be managed, how to identify these Actinobacteria, how to maintain them, and how to utilize them, how to subculture them, and how to optimize these Actinobacteria with particular purpose of enzyme production or other products. So therefore, it is a matter of time only. But working on Actinobacteria is really a great thing in comparison with working on other organisms. Because so many fields are still there, wide scope is there for working on Actinobacteria. So we can isolate many new species. We can find out, we can investigate their role, new role, and we can publish the paper, we can have the patents on these particular Actinobacterial products also. So therefore, this branch is not much new. 20-30 years back, the working was started. And now we are having many more tricks, many more opportunities, many more scope to isolate these organisms. The major obstacle in isolating the actinomycetes is the fungal growth, which overgrow the Actinobacteria. Therefore, to avoid the fungal growth, we have to add antifungal agent, for example, grisofulvin, for example. So you can use another antifungal agent also in media so that the fungal growth can be arrested. But this is trial and error, because it is not like that a single antifungal agent can arrest all the fungal growths. So you have to use combinations also, so that we can avoid the fungal growth. Even we can include other antibiotics to avoid other bacteria other than Actinobacteria. Therefore, selective methods we can use for this particular isolation of the Actinobacteria. Glycerol aspergine agar is one of the medium which is commonly used. Starch casein agar we can use Bennett's agar, or even we can use particular coconut milk agar also. This coconut milk agar, this is our discovery. We have investigated this, Deshmukh sir and myself. We have devised this particular medium, coconut milk agar, coconut water agar, which gives the growth in very less time. So it will avoid the delay. It will avoid the time killing in obtaining the colonial growth on the nutrient media. Because the coconut milk or coconut water contained auxins, which are growth promoters, and with a particular combination, particular composition, particular proportion, if you use this, the growth of Actinobacteria can be achieved in very less time, say 8 to 10 hours or maximum 18 hours. So that is a great achievement. So we can use that, and you can avoid the initial time lapse. And once you have the particular colonies, the main thing is that Actinobacteria, they produce tough colonies. So we can recognize.
Yes, sir.
By,
Sir, please.
Yes.
Sir. Now let other. Miss, again, you will speak.
Okay.
But I want interaction.
Correct.
So, yes, Dr. Sunita.
Okay.
Sunita, what is your research topic?
Okay.
Please tell. And any difficulty any teacher having?
Yes.
Type your question in the chat box.
Okay.
Type your question in chat box. Dr. Sunita will tell about her research topic.
Yes, thank you so much, sir. Like I said, initially I worked on Streptomyces genera, but later on I shifted to rare Actinobacteria because I mean that those genera are the ones, genus are the ones which have not been tapped much. So I tried to isolate several rare Actinobacteria in my work. Right now, the work that I am doing, sir, is particularly to search for antiquorum sensing compounds from rare Actinobacteria. That is my topic right now, to search for antiquorum sensing compounds from rare Actinobacteria. And I have just started this particular project. So I'm isolating these rare Actinobacteria right now, and I'm screening them for their antiquorum sensing activity. The major problem that I face, particularly with rare Actinobacteria, is the fact that if you don't subculture them routinely, they do lose their viability. And in spite of routine subculturing also, at times after a few months of culturing them, they start losing their viability, sir. So this, if somebody could actually help me out with this of how to preserve them long time, because I have many a times lost many of my cultures because of this particular problem of not being able to preserve them. Of course, I have sent many of them to the culture collection centers where they are preserved, but not all. I cannot send all of them. So if somebody could really help me with this problem of preserving them.
Which media you are using for the maintenance of the subcultures?
Normally, I use potato dextrose agar.
Okay.
And I also use yeast extract, malt extract agar. That is ISP2, International Streptomyces Project Medium 2.
Okay. You can
Yeah.
try glycerol aspergine agar, madam.
Okay, sir. Okay,
Sir,
sir. I will
madam,
try.
ma'am, sorry for the interruption.
Potato agar is mainly used for the fungal growth.
No, sir, we are getting very good activity growth on PDA also. For mass cultivation, yeah. When I go for mass cultivation, I go for PDA, that is.
Yeah, you may get growth on
Yes,
the
madam.
PDA, no doubt, because it
Yes.
is a rich medium. But
Yes.
if you try glycerol aspergine agar, then you can maintain for longer time. No
Okay,
doubt.
sir, I'll try it. I'll try it.
Ma'am, what is the common contaminant, ma'am, that you are getting?
Like sir
Most
said,
common
yeah, like sir said, if we don't add antifungal compounds, because
Yes.
these are slow growing, actinobacteria basically are very slow growing. So by the time they grow, many a times the other contaminants, particularly fungus, fungi, and many a times even the fast growing soil bacteria like bacillus, very common contaminant is bacillus.
Yeah.
Correct.
Yes. But like Kulkarni sir said, I will definitely try coconut milk agar to get faster growth.
Yeah. Definitely you will get good results, no doubt.
Yeah, thank you so much, sir. I'll definitely try it. Yeah.
Faiza Ibrahim. Could you please introduce yourself? Yeah.
Excuse me. I'm Dr. Shivakumar.
Yes, sir.
Yeah, coming to preservation, I think if you are going repeatedly for subculturing of actinomycetes, it will lose its viability. So I can suggest you to just go for 30% glycerol stock. So that, but every 1 year you have to retry the culture and you have to check the viability. Or otherwise you can go for lyophilization. These are all the method for preserving cultures actinomycetes for a long term. Thank you.
Thank you. Shivakumar sir.
Yes.
Could you please introduce yourself, sir, because we would like to hear from you.
Yeah, I am Dr. Shivakumar from Center of Advanced Studies in Marine Biology from Annamalai University.
Glad to hear from you, sir.
Yeah,
thank you.
I contributed especially on marine actinomycetes.
Thank you so much, sir.
Hello.
Yes,
I'm requesting all microbiologists working on actinomycetes, please write your email ID in chat box. Everyone,
yes,
because other participants can ask you question on your email.
Yes, sir. That would be helpful. Kindly enter your mail ID. Kindly type your mail ID and even contact number. It would be very useful, sir, if you give us a contact number also.
Yeah, thank you. I am driving. I cannot. But anyway, Ocean Actino, it's my Gmail is my mail ID. I'll try to just type it.
Okay, sir. Okay. Yeah. Thank you, sir. Saravanan, sir. Dr. Sunil Kumar, could you please start your discussion? Sunil Kumar, sir.
Yes, sir. Yes, ma'am.
Yes, ma'am. Okay, you are on behalf of, may I know your name, ma'am?
Chandigarh University. Dr. Sunil, I am. Actually, my voice looks like, like female. My voice
okay, I'm so sorry. It's okay, ma'am. Please start your discussion, ma'am. We would like to hear from you.
Yeah, we are just working initially right now. I have only one student doing master under my supervision here. So we are just going to find out like any actinomycetes there in the the sample of the people, the hand samples. So we are just taking their sample with the help of swab and we are culturing. We cultured last week. So here the lab is new. Everything is new. So we haven't any student are before this, right? So that's why we are looking at maybe we'll get something better results. So we are working on it. We are also finding the resistance bacteria because we are going to check the resistant pattern of the organism. Is there any drug resistance which is going to be spread in the future for maybe any problem is there, right? So up to species level, we are going to identify with the help of Malditov. We'll collaborate with PGI also because I did my PhD from there. from PGI Chandigarh. So we'll try to like collaborate with them so they can also find, give us some permission to identify up to the species level. So we can just, we are working initially. We started recently. So let's see, ma'am, if we'll get anything new or any results come out, then we'll definitely will share with the all the people.
Excellent, ma'am. Thank you so much. Aparna, Miss Aparna,
Yes, ma'am.
could you please share your scientific ideas on actinomycetes?
Yeah, ma'am. Right now, I'm a PhD scholar. I already told about that previously, that I am working under Dr. Bhaskar Rao sir in VIT campus, Vellore, Tamil Nadu. My topic is also actinomycetes. Just now I started my research, so many doubts are there. That's why I just want to join this meeting. That will be helpful for my further research. So I isolated few of isolates from environmental sample, but still it's just now started. So I don't have much idea, but I just want to know many things, and many good people are there. They are giving so many good ideas that will be helpful for my research in my PhD.
Thank you so much. All the best.
Thank you so much, ma'am.
Miss Vandana Kumari, could you please introduce yourself? You have just joined. Could you please introduce? Vandana Kumari.
Yeah. Good afternoon, ma'am.
Good afternoon.
Ma'am, I am working in biomaterials only, not working in biology. Just I want to interested in for knowledge.
Okay, just get the ideas, right?
Yes.
We're glad. All the best. Okay,
Thank you.
thank you. Miss Loveleen.
Good
Miss
afternoon.
Loveleen. Yeah.
Good afternoon, everyone. I'm a second year student, and I'm doing bachelor's in microbiology from Chandigarh Postgraduate Government College, and I have also joined for knowledge.
Yeah, you are a budding star of microbiology. Okay,
Thank you
all the
so-
best.
Thank you so much for all of your knowledge and time.
Yeah, good luck. Miss Swati, could you please share your ideas? If you're a student, if you're willing to acquire the knowledge, just share your thoughts and ideas. Swati? Any questions or any queries among yourself? Could you please put it in chat box, or you can also speak out. You just unmute and please share your ideas. This is a live interaction.
Yeah, ma'am. Actually, I just want to know that any of us, like whoever is in meeting, worked on extreme environment, actinobacteria, like hot spring or some high temperature environmental sample.
Yes, Dr. S.W. Kulkarni will answer it.
Yes.
Let's see.
So extreme environments include the particular hot temperature mainly, and you can also isolate from salts also. That is the sea sample. That is also an extreme environment. Basically, these two you can try. On which sample you are using, madam?
Sir, right now I am using some hot spring sample or marine as well. So
Okay.
it's not yet decided.
Okay, no doubt. For this particular, you have to collect the sample and maintain the temperature mainly. Even when you collect the sample and transport to the laboratory, and in laboratory also you have to maintain that temperature, and that means we have to give equal temperature or the same temperature until you get the growth.
Okay, sir. Sir, one more doubt is there. For production medium, is there any specific media to use so that they can able to produce some specific and helpful enzyme for some further application?
For particular enzyme isolation, you have to add particular substrate in that. So you have to avoid other minimal medium you have to use, and you have to add particular substrate so that you can test whether that actinobacterial isolate can produce that enzyme or not.
Okay, sir. Like some marine sample, people are using SS medium. I saw some of my colleagues. But for terrestrial sample, as of now, I don't know. Like, I mean, what I should add or how can I make some production medium that will be helpful for extracting some enzyme like that. I'm just want to know.
You can use basically that glycerol asparagine agar is the most common. Coconut milk agar is there. Starch casein agar is there. You can use any medium and you can assess the particular enzyme production.
So starch casein broth will be also like I can use as a SS. I mean a production medium.
Yes, yes.
SS broth, starch casein broth. In that also I can try to grow them and get some enzyme related things later on. I can screen that whether they are producing enzyme.
But you will get crude enzymes or combinations. Then you have to purify that.
Yes, sir. Later on, I will purify. In crude level, I will just check that whether there is some activity or not. If I will get means, I can go for other purification, extraction, and other things.
Correct.
Okay, sir. Thank you so much, sir, for this information. Thank you.
Adding to his point, Sarah's expert has rightly informed the information to you on just including the substrate. When coming to the extremophilic farms, we have experience work with the ontotic samples.
Okay, sir.
And we just work for psychrophilic actinomycetes.
Yeah.
Okay, sir.
And we found the farms Saccharopolyspora and again Nocardiopsis and Streptomyces. These are all the common farms. And we also done a metagenomic studies of the ontotic waters, especially pertaining to actinomycetes. This is coming to ontotic, extremophilic.
I know, sir. I heard about that. Some researchers are working on this because it's like two ice. I mean, minus temperature is there. In that also, they are able to survive in such habitats.
And again, coming to halophils, if you go for mangrove ecosystem, there is salt marsh area is there.
Yeah.
Yes, sir.
It's predominantly occupied by a plant species Suveda. And if you just get the sample from there, you can get the actinomycetes which is capable of growing in 20% NaCl. You cannot imagine 20% NaCl. So commonly you can get Actinopolyspora from saline water. That is saline instance, sodium dominated ecosystem, that is hypersaline ecosystem, halophilic. And if you are working for thermophilic, again we have the experience from volcano, from Andaman. And one of my scholar, Dr. Arti, studied from volcanic samples. She got a lot of, again, Saccharopolyspora from the volcano, along with Streptomyces.
Yeah.
Okay, sir. Can I get? Sir. Can I get any research paper? Like, she published some research papers which I can go through and understand.
You just put it. Ontotic actinomycetes, metagenome Shivakumar, you can get my document.
Okay, sir. Thank you so much, sir.
Also, you can get, if you type my name and volcano, you can get, with actinomycetes, you can get the document. And Actinopolyspora, we discovered a new species, an Actinopolyspora indiensis.
Okay, sir.
And
Nice.
species identification, lot of information are there. If anybody is interested on particular aspect, I can deliberate on that. Thank you.
Thank you so much, sir, for this such nice information.
Any other discussions?
Yes, one thing I want to interrupt. Hello.
Yes, sir.
Yes.
I want to make one request to all microbiologists working with the actinomycetes. In India, there is not a single depository for Actinobacteria. we have depository for various types of microorganism, various groups of microorganism. So any one of you can apply to the DBT and can ask for a grants funds for depository. I'm sure the DBT will definitely sponsor. For that purpose, you have to show your research in actinomycetes and a few cultures which are identified in your lab. So many workers are there in India. They can think in this manner. Thank you.
Yeah, already in InTech, that's a small portion of actinomycetes group has been maintained in InTech, Chandigarh. And
Yeah.
Along with that, there is also in MCC Pune, they are also maintaining actinobacteria
Correct,
cultures,
correct.
cultures.
But there
I
is
think
no
headed by Tapan Chakrabarti. To preserve the marine actinomycetes, there is a culture collections in Cochin. That is in NIO regional center. There is one person maintaining marine actinomycetes. These are all the things. But as he said, if you are specifically interested, you can also just write a proposal for DBT for maintaining the culture collections for actinomycetes. That is a very good idea.
Instead of scattered centers, you can
Correct.
have a centralized center somewhere.
Yeah, that's correct. If you are, there is for fungi, there is Agarkar Research Institute is having a premier center for maintaining all the fungal, fungal cultures. And like that, for cyanobacteria, NFMC is there, National Facility for Marine Cyanobacteria. Like that, exclusive center should be needed for preserving actinomycetes, and it's a very important thing for our country.
very true. Now,
Any other discussions,
again,
sir?
I request Dr. S.W. Kulkarni or Dr. Sunita ma'am. The question was, it should be called as a actinobacteria, not actinomycetes.
Yes, yes.
It's a very
Actinobacteria
true.
nowadays.
But why? And one more question, my question. If you can give literature, books, journals available for the actinobacterial knowledge, Burgess manual, can you focus on it?
Sir, we are having one manual that we have prepared during the workshop. So we can circulate that also.
Correct. You tell it.
Yeah.
Yes.
So it covers all the introductory part and also the particular methodology which is used for the isolation and identification at any lab level. Even though you can have minimum equipments, you can identify to some extent, and you can on the basis of morphological, biochemical, and cultural characteristics, you can go for the particular species level to some extent, and then you can molecular level you can try somewhere else. But basically, that manual will cover all the particular aspects regarding the study of actinomycetes as such. So we are having that. We will circulate as per the demand, no doubt.
Yeah, rightly said. And apart from, along with Burgess manual, as he mentioned, the expert mentioned, 70% of the farms are predominantly streptomyces when you are going for isolating actinomyces.
Correct.
That is true. So like that, there is one Hido Nanamura from Japan. He prepared a key for identification of 535 streptomyces. So I think that will be a very valuable information.
Yes.
That is having the spore chain morphology, spore surface morphology, and what are the carbon source utilization pattern, and what are the pigments like soluble pigment and riverside pigment. these are all the detail. So if you want to just identify in the preliminary level up to species level, only do this. Go for scanning electron micrography. Just get the things done for spore chain morphology and the spore surface. Then go for pigment pattern and go for carbon utilization pattern. Then having the result in your hand, compare the result with the species given in the Hidononomura key. It is having around 550 around streptomyces. Just compare it and identify the species. If not possible, if your species not fitting in the Hidononomura key, then I suggest you to go for polyphasic taxonomy for identification of the species.
Yes, yes, very true, sir.
Sir, I prefer to call it Actinobacteria, and I think almost everybody now should be calling it Actinobacteria, because like these organisms are, they used to be called fungi-like organisms, fungi-like microorganisms, and maybe that is why actinomycetes. But then it creates a lot of confusion, because once when I was presenting my work, there was an eminent scientist, and because I was saying actinomycetes, he did not accept that I had identified it using 16S. He was like, why did you not go in for 18S? So I had a tough time trying to explain it to him. And from that time onwards, in all my research papers and my project proposals, I have shifted to Actinobacteria. Burgess also now calls it Actinobacteria. And like Shivkumar sir just now said, I would just add a little bit about isolation of rare Actinobacteria, because I'm working on it. Yes, most of the actino that you land up are mostly streptomyces. But if you give specific pre-treatments to the samples, soil samples or water samples, and if you add take up very specific medium and add very specific antibiotics to it, you land up with very specific genera of rare Actinobacteria.
Correct. Very true. Very true.
Yeah, that is a very good Nobel lecture by Hayakawa. And in that, he has explained beautifully about the different pre-treatment methods and the different antibiotics, which when added can give you. So different treatment and different cultivation methods can give you very specific rare Actinobacteria. So at least to some extent, we can say that, yes, I have isolated Micromonospora. And then like Shivkumar sir said, using the spore morphology and the spore surface morphology and the other patterns which are given in the volume 5 of Burgess manual, we can, yes, to a great extent, even without molecular characterization, come to the conclusion of the genus and then go to for the molecular characterization.
Yes, that's correct.
Okay, thank you all. Now we had a very fruitful discussion. And one more thing, everyone has written their email ID and phone numbers. So other research workers interested in the research of Actinobacteria, because Burgess manual has fully accepted it as a Actinobacteria now. So I request everyone, come together for the research on Actinobacteria. I will request Dr. S.W. Kulkarni to circulate the book he is having on Actinobacteria, correct?
Yes, definitely.
And, yes, and we'll work more. If there is any question, later on you can ask to the concerned teacher via mail. And I request Usha Shri ma'am again conclude this group discussion. Thank you. Usha Shri.
Yes, sir. Good afternoon, everyone. I take a great pleasure in concluding this great scientific event. And I thank one and all for sharing your noble ideas. And it would be very useful for everyone, especially for budding microbiologists who have joined this meeting. I thank Dr. A.M. Deshmukh sir for providing us an opportunity to learn so many things about Actinobacter. And surely, sir, we will further proceed in developing the research by sharing the great and novel ideas, by performing so many experiments using novel bacteria, that is Actinobacteria. Thank you all. Have a wonderful weekend.
Thank you.
Thank you all.
Ushashri, please
So
end the
Deshmukh,
meeting.
sir.
Please
Yes, sir.
end the meeting.
Yes, sir. Thank you so much, sir.
Thank you,
sir.
ma'am. Thank you so
Thank
much,
you
sir.
all. Have a nice weekend. Thank you all.
Thank you. Thanks, so. Thank you.
Thank you,
Thank
madam.
you. Thank you, sir. Thank you.
Ushashri,